العافية. Thank you all for being here today and joining me to welcome Professor Colin Taylor. Professor Colin Taylor, he finished his natural science degree at Gonville and Caius College in Cambridge. Then completed his PhD in insect physiology. After that, he did postdoctoral work in the USA before returning to Cambridge as a research fellow at St. John's College and a Royal Society Lock Research Fellow. He was appointed to a lectureship in pharmacology afterward, and then he was appointed professor in cellular pharmacology in 2001. Professor Taylor served on various editorial committees and funding agencies. Please, one's silent. Professor Colin is a member of Academia Europaea and a fellow of the Academy of Medical Sciences, and he's also a Wellcome Trust senior investigator. Professor Taylor's lab typically include about 12 staff from different nationalities, including postdoctoral, lab manager, PhD student, and visitors. And we are fortunate here in Al-Quds University to have the opportunity to host Dr. Taylor, Professor Taylor, to learn more about his research. So the floor is yours.
Thank you very much. And sorry about the delay in getting started, a bit of incompatibility with the laptops there. So this talk might, in that case, be as much of a surprise to me as it is to you, because I'm not quite sure which talk it is I've uploaded here onto the alternative laptop, and it's possible we may not get the movies. So let's see how we get on. So as you can see from this slide, there's a lot going on there in the outside world. And although you may think that it's Google and perhaps worse still, Fox News that controls your access to that information, the truth in fact is rather more prosaic in that the real barrier to information transfer is this membrane, the plasma membrane that surrounds every one of your cells, all 10 to the 13 of them in your human body. And this membrane, just 2 molecules thick, is the barrier across which transmit information if our cells are to understand what's going on in the outside world, and to understand also what the other 10 to the 13 cells are doing in your body. So the core of the problem then is to get information across this lipid bilayer. I'm not having much success with the across this lipid bilayer to the inside world of the cell. And for the most part, that's the job of proteins, receptor proteins embedded in that plasma membrane that sit with one side poking out into the outside world and the other to the inside world. And when these receptor proteins bind a signal in the outside world, they change their shape. That then is detected by proteins at the intracellular surface of these allosteric receptors, and that then communicates onwards to regulate cellular activity. And one of the means by which that signal is passed onwards to recognize cellular activity is through the intracellular calcium ion here, which as you can see is an element. So the only thing we can do in terms of allowing calcium to serve as one of these intracellular signaling messengers is to change its distribution. We obviously can't make or degrade an element. So cells invest considerable energy in excluding calcium from their cytosol into the outside world by pumping it out across the plasma membrane, and into intracellular organelles largely by pumping it into the endoplasmic reticulum, and that leaves the cytosol of the cell very low, around 100 nanomolar or so, relative to the millimolar or more outside the cell and the hundreds of micromolar within the ER membrane. So those membranes then are rather like the dam here, holding back this tide of calcium ready to invade the cell. And just as we use the sluice gates in the dam to control that flux of water through the dam, then so we control the flux of calcium into the cell by putting a gate into one of these membrane proteins, and normally the gate is closed, but then when we open that gate we allow calcium to flow rapidly down these concentration gradients to give us the cytosolic calcium signal that goes on to regulate so many things that the cell does. And as I've hinted already, these channels, these calcium channels, exist in different membranes. They exist here in the plasma membrane and in the endoplasmic reticulum, and 2 of them and the interactions between them because the interactions between these channels are very important throughout biology. The two that I'm going to spend some time talking about today are primarily, I'm going to look at the Ipd receptor, which is an intracellular calcium channel resident here in the ER, and then I'll come back toward the end and say a little bit about a channel in the plasma membrane, the ORI channel, that ultimately ends up being regulated also by the Ipd receptor. So these interactions between channels in these different places then are important as a means of generating cytosolic calcium signals. But, oops, we seem to have snarled now on the computer.
Okay.
There we go. But one question I think that we need to ask is that given that these channels in the plasma membrane evolved before the ones in the ER, what is the selective advantage to a cell of having evolved all the machinery that allows cells to accumulate calcium into the ER and then regulate its release from it? And there are a number of arguments I can put for that. The first is this, is that both the calcium pumps here in the ER and in the plasma membrane pump calcium across a concentration gradient out of the cytosol to the much higher calcium concentrations on the other side of the membrane. But at the plasma membrane, there's also an electrical gradient, an unfavorable electrical gradient, so that calcium has to be pumped against that additional barrier, and that costs more. And it costs more to the tune of the fact that we can pump only one calcium ion for every ATP hydrolyzed at the plasma membrane, where we can pump two for every ATP hydrolyzed into the endoplasmic reticulum. So the upshot of that is it's cheaper to shuffle calcium to and fro out of the ER than it is to shuffle it to and fro across the plasma membrane. The second thing we need to think about is that at the plasma membrane, there are many other ion gradients across the plasma membrane, themselves doing all sorts of important things, sodium and potassium gradients notably, such that if we are to open a gate in our channel and selectively allow calcium to pass, we are going to have to select between cations as the calcium passes through that channel, so as not to just collapse all the other gradients that are doing all these important things. And the means by which we achieve that usually is to put a narrow selectivity filter into the channel so we can taste the ions as they pass through and reject the ones that we don't want to allow to pass. But that takes time, so that the opportunity to select calcium comes at the cost of slowing the rate at which calcium can pass through the channel. So at the plasma membrane, our calcium selective channels tend to have a rather low conductance. But in the ER, the only cation with a significant gradient across the ER membrane is calcium, so we don't need to be very selective as the ions move through it. We can have a very wide selectivity, and calcium can gush through the channel when it's opened. We can have very large conductances, and so generate very large local calcium signals around these intracellular calcium channels. And the third argument is clear here, which just shows you a HeLa cell in which we've stained the ER, we've labeled the ER with a GFP targeted to the endoplasmic reticulum, and you can see it forms this beautiful reticular network that invades the entire cell, and that means that these Ipd receptors are sitting distributed throughout the ER, ready to deliver their calcium to all sorts of places, locally delivering it to the cytosol, and perhaps even more importantly, delivering it also to other intracellular organelles that I'll say a little more about later. So we have this distributed delivery system, as it were, for calcium through the endoplasmic reticulum, often maintained by junctions between proteins that stabilize junctions between the endoplasmic reticulum and the intracellular organelle with which it's interacting. And I want to say just a little about some of the work from my lab that's looked at the interactions between the endoplasmic reticulum and the lysosome, the principal degradative organelle in all eukaryotic cells. And the starting point for those observations is shown here. This is a human embryonic kidney cell, but it's the same in other cells, where the cells have been loaded with a calcium indicator so we can monitor the cytosolic calcium concentration. We then stimulate the cells with a ligand that activates one of these receptors. It's carbachol activating a muscarinic receptor. So that gets the message across the plasma membrane. It makes the IP3, and as you can see from the black line, it then releases calcium from the intracellular stores through the IP3 receptor. The crux is that when we inhibit the VATPase, the proton pump of the lysosome using perfilomycin, we get much larger calcium signals, and you can see that across the whole dose-response curve. It's the same result if we use siRNA to knock out a critical subunit of the VATPase. These details don't terribly matter. The upshot is, if we use other means of perturbing lysosomal function, this is a peptide that can disrupt the lysosomal membrane. The result is the same. We exaggerate the cytosolic calcium signals. And we can also use a third treatment, vacuolin, which just enlarges lysosomes without actually damaging them, and the story remains the same. The cytosolic calcium signals are bigger than they were before we inhibited the lysosomes. Now, I won't rehearse all the evidence with you because it's all published, or most of this is published. Suffice to say that the reason for that is that normally lysosomes are sequestering calcium that's being released from the ER, so that when we inhibit the lysosomes, more of that calcium makes itself evident in the cytosol than was hitherto the case. So that's all calcium released from the Ipd receptor from the ER, but I mentioned a moment ago that there is another means of calcium entry across the plasma membrane. I'll say more about it later, and that's this store-operated calcium entry pathway. And if we activate that pathway, so now calcium is flowing in the cell across the plasma membrane, you can see that that gives us nice big cytosolic calcium signals, but they're totally unaffected by disrupting the lysosomes. And again, the details down here don't matter. Suffice it to say that it doesn't matter how we activate that store-operated calcium entry, the result is the same. The cells don't care about calcium, the lysosomes don't care about calcium entering the cell across the plasma membrane. They're only interested in the calcium that's being released through the Ipd receptor, and that's the calcium that gets sequestered by the lysosome. And that fits, I don't think this will work, this is a movie, but no, I'm afraid we're going to miss out on the movies today. That fits with these observations, which would have been a movie of the ER here and the lysosomes shown in red. And what you would see if the movie was running is that the lysosomes crawl along the endoplasmic reticulum, maintaining this contact between lysosomes and ER.
Okay. You just go to this one.
This one?
Yeah. Yes.
Okay. Now, all that I've shown so far has used pharmacology to disrupt lysosomes, and we wanted to try and see whether we could develop the story by using a non-pharmacological disruption of the lysosome. And the way we did that was to take advantage of the fact that we also have calcium indicators encoded by proteins, and one of these is this protein gecko, and we can then hijack the cell's address machinery that it normally used to send proteins to appropriate destinations, so we can attach a tag to these proteins and send them to various destinations. So what we did is we made a very low affinity calcium indicator. Remember, normally the cytosolic calcium concentration is around 100 nanomolar, and this is very much higher, so it's not interested in low calcium concentrations. And then we expressed it either on the surface of the lysosome to monitor what the calcium was doing around the lysosome, or else we expressed it in the cytosol. And again, the movie isn't going to work, I'm afraid, so we'll have to skip the movie. Then when we record these calcium signals evoked by activating these HeLa cells with histamine to make IP3, what you see is around the lysosomes, and this just shows you the fluorescence intensity we record from this calcium indicator relative to the number of these punctate particles that we see, the lysosomes within the cytosol. And what you see is that at the top end here, we have about half the lysosomes that detect a very high calcium concentration. You can compare that with the cytosol here, where all the cytosol is seeing is down at this bottom end, and some of the lysosomes see that as well. But so they're the lysosomes that seem to be no different from what the cytosol experiences, but about half of them are clearly being exposed to a very much higher calcium concentration than the rest of the cytosol sees. And that's the result with histamine, which remember is making IP3. Here's the lysosome reporter, and here's the cytosolic reporter. And if we do the same with our store-operated calcium entry pathway, you see the cytosol and the lysosome report the same thing, reinforcing that story that I suggested a moment ago, namely that now if we put the calcium sensor on the surface of the lysosome and we don't do any disruptions, it's confirming our story that the Ipd receptor selectively releases calcium that is seen by the surface of the lysosome, whereas the same or even more calcium entering across the plasma membrane, the lysosome has no interest in that calcium. So that's summarized here, just showing you that calcium released from the IP3 receptor is selectively accumulated by these lysosomes, and that presents a rather close analogy with mitochondria, which people have been working on similar ideas for many years, where mitochondria also selectively accumulate the calcium released near to them by ion channels, and again often the IP3 receptor. So that means that both mitochondria and lysosomes can really shape these cytosolic calcium signals by sequestering some of the calcium that's released. The other thing it means, and this is best developed for mitochondria, is not only are these organelles shaping the cytosolic calcium signals, but the organelles are themselves responding to this local delivery of calcium to the organelle. So that in the case of the mitochondria, this calcium uptake arising from the activity of the IP3 receptor regulates their mobility, it regulates oxidative phosphorylation, for example. And in the case of the lysosome, there's evidence too that it might regulate the activity of some of the lysosomal channels and regulate trafficking within the endolysosomal pathway. So this selective uptake then of calcium released by IP3 receptors serves not only to shape the cytosolic calcium signals, which most people pay most attention to, but it also regulates the activity of the organelles that's doing the sequestering. So coming back then to the, as I say, slightly different talk to the one I was anticipating, coming back to these pathways that link plasma membrane receptors, these proteins that get signals across the plasma membrane to the IP3 receptor. You can see here are these families of receptors. They, when they're activated by the extracellular stimulus, stimulate an enzyme here that degrades the lipid PIP2 to give us IP3, and that, as we've been seeing, goes on to activate the IP3 receptor and open that gate in the channel analogous to that sluice gate in the dam. But in fact, IP3 alone is not sufficient to cause the opening of an IP3 receptor. What IP3 does is to prime the IP3 receptor such that having bound IP3 it can bind calcium, and calcium then causes the channel to open and release calcium into the cytosol. So we need two signals, IP3 and calcium, to open the channel. And the importance of that is it means that we can get this regenerative phenomenon whereby calcium released by one channel can ignite the activity of a neighbor if it has IP3 bound to give us this calcium-induced calcium release, and so propagate calcium signals regeneratively. So in a sense then, IP3, in this amplifier analogy, IP3 is the regulator that sets the gain on this calcium sensitivity in the IP3 receptor, so how regenerative the signals will become. But the other important element of it, of course, is that the geography matters too, because if two IP3 receptors are very close to each other, they're more likely to feel the calcium that's spewed out from a neighbor than if they're further apart. So both IP3 and the geography, the distribution of the IP3 receptors, play important parts in determining how regenerative these signals become. And we can see that here, where I'm just going to show you the consequences of moving up a stimulus intensity. This might be histamine, delivering more IP3 to a cell, for example, as we increase the concentration of histamine. At the bottom of the curve here, we can detect with microscopy single IP3 receptors open, giving us very local calcium signals. As we move further up, we find the calcium released by one IP3 receptor spews it over its neighbors, and if they've got IP3 bound, we'll get opening of a cluster of IP3 receptors, and that gives us a calcium puff. And again, I'm afraid we're probably not going to get the video. No, so we'll have to skip that. Further up, the calcium spreading from one of these calcium puffs to a neighbor gives us a calcium wave that spreads across the whole cell. And then at the top of the curve, those calcium waves become more frequent as we further increase the stimulus intensity. So we move up this regenerative propagation of the calcium signals then as we move up the concentration effector curve, the dose response curve. Again, all determined by the fact that IP3 is dictating the calcium sensitivity of the IP3 receptor, and that the geography of the IP3 receptors is dictating whether they're going to feel the calcium released by their neighbors. So both are important. And this geography really is what spawned the work underlying the final part of my talk, and that is we were interested, because of the importance of this geography, in trying to measure the behavior of IP3 receptors, that is the calcium they're releasing into the cytosol, at the same time as we report the presence of the IP3 receptors in the cell, so that we can see the proteins at the same time as they're releasing calcium into the cell, to try and see directly how this geography matters. And to do that, I don't need to dwell on the details. We used a gene editing method. We happened to use Talen's. The details don't matter. But what we did was to attach EGFP, a green fluorescent protein, to the N-terminal of the endogenous IP3 receptor of HeLa cells. So we're not overexpressing the protein. This is protein expressed at the native level. The native protein now carries with it an EGFP tag, so we can see exactly where the native protein is in these cells. And again, this is just to validate the method that we've used. The first point just shows you that the only protein that's tagged in these cells from this fluorescent gel is the IP3 receptor. This one, a Western blot, shows us that all of the expressed IP3 receptor is EGFP tagged. This shows us that the tag protein is functional, because if we treat our cells with a siRNA to EGFP, we attenuate the calcium signals that we can evoke in those cells. And alternatively, if we take the protein that we've expressed in those cells, the EGFP tag protein, and make an exact mimic of it, and then express it in another cell that doesn't have any IP3 receptors of its own, then those cells become competent to respond to IP3. So it tells us that the tag protein has not lost its ability to release calcium. And then finally, you see down here that these green particles identify our IP3 receptors in these HeLa cells, and the red reticular structure is the ER that we've labeled with an red ER-targeted protein. Again, we're probably not going to have the benefit of the next slide, which is a movie. Yeah. So what we use then is total internal reflections microscopy, TIRF microscopy, which allows us to look at a very thin sliver of the cell, just about 80 or 90 nanometers thick, into the cell and get a very high resolution image of what lies in that few nanometers beneath the plasma membrane. And if the movie were running, you would see here's just a corner taken out of a cell showing you the ER network in red, and dotted around it are these green puncta, which are the EGFP tag native IP3 receptors. And what you would see is that many of these IP3 receptors are whizzing along the ER network, and others of them are immobile within it. Now, this TIRF microscopy allows us to look only at a thin sliver immediately beneath the plasma membrane, but we can go with confocal microscopy and take sections across the whole cell, and this just shows us those sections starting here at what would be the equivalent of the TIRF field adjacent to the cover slip, and going all the way through the cell till we get at the top surface out here. The important point is that no matter where we look in the cell, we see this similar punctate distribution of these endogenous IP3 receptors. They're everywhere in the cell. Our next question, though, was to ask, but what is one of these green spots? How many IP3 receptors are there within one of these green spots that we see in these gene-edited cells? And for that, we use single-step photobleaching, where we turn up the laser power now and try deliberately to bleach the fluorescence, and then we see the steps as we bleach the individual fluorophores. And that leads us with some assumptions to tell us that each of these green spots that we see resident within the ER contains on average about 8 of these IP3 receptors. Some contain many more and some contain fewer, but the average is about 8 tetrameric IP3 receptors in each of these green spots that we see in the ER. We can go to another super resolution method where we get better resolution still, and that's STORM. I won't dwell on the details, which then allows us to identify individual fluorescent spots within those puncta. And the important message from that is if we just look here, here's our TIRF image. You can see it's rather fuzzy because of the diffraction limit of a normal microscope. But then when we see the TIRF image, the STORM image, you can see that it sits within that green image, but now we can see these distributions of IP3 receptors within those puncta. The message I want to lay before you here is that within those puncta, we have quite widely distributed IP3 receptors. The little yellow particles I've drawn in the corner here show to scale the size of a single IP3 receptor protein. And you can see that many of these IP3 receptors are too far apart to be physically holding hands with each other. And there must instead be some sort of scaffold protein that is holding these IP3 receptors together within these clusters that maintain this this assembly of these clusters as they move around the ER. So we'd of course dearly like to know what that scaffold protein is that organizes these IP3 receptors into these fundamental units that we think of 8 or so IP3 receptors in each of these structural units. Now, I didn't have the opportunity to show you, but I've mentioned that many of these IP3 receptors are mobile and a few aren't. And so we can track now the individual trajectories of these IP3 receptor puncta to look at their behavior. And we did that for about 3,000 of these puncta, and you can see when they do that, they beautifully trace out the reticular network of the ER. So every one of these tracks just shows one IP3 receptor punctum and the path it took over a few seconds, and collectively they just define the ER, much as we would expect. Now, if we then plot the mean squared displacement, that is how far the particles have moved in unit time, we would expect if they were moving by diffusion, that line would be linear. The fact that it curves upwards tells us that some of these proteins are moving by directed motion. They're being actively moved through the ER rather than just moving by diffusion. And we can see that here. This shows us an IP3 receptor punctum that didn't move at all. This one shows us one moving randomly by diffusion, and here's a typical example of an IP3 receptor that's moved along one of these directional trajectories. And if we just categorize those, you can see that only about 7 or 8% of them move directionally, most are moving by diffusion, and about a third are immobile over many, many minutes. Turning to the directionally moving ones, we noticed from co-localization studies that many of the IP3 receptors in green here overlaid the microtubular network in these cells. You can see here these IP3 receptors all aligning quite nicely with the microtubules in these cells. So we asked then whether the microtubule motors might be responsible for moving these IP3 receptors along the microtubule network. And the short answer is yes, they do. I won't dwell on the details, but if we inhibit the dynein and kinesin motors, then you can see that we lose almost all of those directional trajectories. There's another trick we can use, but it's probably not going to work very well without a movie. And that is we can use this artificial means of dimerizing two proteins through rapamycin. And the way we do that is we attach to our motor a protein, FKBP. So this is the kinesin motor here, and it's now got another protein tagged onto it, FKBP. Details don't matter. We then have another protein which we target on the plasma membrane, which has FRB tagged to it. Again, the details don't matter. What does matter is that when we add rapamycin, that causes FRB and FKBP to dimerize with each other to form a very tight association. So what that means is that if the kinesin motor walks along the microtubule, as it does, towards the plasma membrane, that's the usual direction of motion of a kinesin motor, it carries its cargo with it, and that's fine, it's just behaving normally. But if when it gets to the plasma membrane, we have rapamycin there, it will get locked at the plasma membrane and locked there with its cargo that it's carried. And as I said, this isn't going to work, I'm afraid, without the movie. But this is the kinesin, this is EGFP-IP3 receptor. If I were able to show you the movie, you would see that when we add rapamycin, the kinesin motors collect out here at the periphery of the cell, and the IP3 receptor collects with them at the same places, showing that the IP3 receptor is being carried by the kinesin motors. So the take-home from that, then, is that this small number of IP3 receptors that move by directed motion do so by moving by these microtubule motors along microtubules. Now, we can look at the immobile IP3 receptors with a trick I've shown here. So what we've done in this slide is to take an image of our cells, and here they are shown in the native green. This is the IP3 receptors. And we take another image 30 seconds later, and we pseudo-color it purple. They're all green, really, but we just use the image processing to make them look purple. So they're just 30 seconds apart. And now if we overlay them, anything that moved in those 30 seconds will be a green or purple, because they'll just be the one non-overlaid color. Anything that overlays that didn't move in the 30 seconds appears white, because green on top of purple gives you a white image. So green and purple are the moving IP3 receptors, and white are the ones that didn't move in the 30 seconds that we chose to look at them. Now we can use a photo bleaching strategy to ask, is there any exchange between the mobile and the immobile IP3 populations? How independent are they? So we bleach a wide strip across the cell, and then we look to see how quickly the fluorescence recovers within that bleached area. And I think what you can see is we start off here with lots of white in the bleached area. It's just enlarged at the end here. And here, even 15 minutes after the recovery of the bleaching, you can see lots of purple and green, but there isn't any white. That's enlarged here. Here's the pre-bleach, all three colors. But then in the post-bleach situation, we get the mobile IP3 receptors detected, but we don't detect any of the immobile ones. And that's quantified down here. Here's the rapid recovery of the mobile IP3 receptors and negligible recovery over really very long times of the immobile IP3 receptors. So what that tells us is that there is really very little exchange between the mobile and the immobile IP3 receptors, and that despite the fact that I've suggested that these puncta are really rather loose confederations of IP3 receptors. So the two populations then seem to lead rather independent existences with no ready mixing of the mobile and the immobile IP3 receptors. Now going back earlier, you'll remember that although we focused in the turf field because we get the most beautiful images, that we see these puncta in the turf field, but the confocal microscopy told us they were present throughout the cell. So we can, again, this movie isn't going to work, so I won't show you it. We can look now to see where did the calcium signals happen. Oops. No, there's a slide. It's skipping a slide here. Okay, I'm going to have to tell you something, and that it's lost something. So two messages. If we look, the first one is if we look with confocal microscopy to see where the calcium signals are happening, we see that all the calcium signals happen near the plasma membrane. And it doesn't matter whether we use endogenous signaling pathways like the histamine or the carbachol I've been describing, or whether we use photolysis of caged IP3, so that uniformly releases IP3 throughout the cell. In both cases, the only places that these calcium puffs that I mentioned to you a moment ago initiate is immediately adjacent to the plasma membrane. So that allows us to go back to the turf field and say, well, if all the calcium signals are happening near the plasma membrane, we can follow them by using the turf microscopy and ask what's the relationship between the IP3 receptors and the calcium signals that they generate. And this just shows you 3 or 4 of these calcium puffs detected with the calcium signal, and here's the localization of the IP3 receptors. And the take-home message from that is, here's just 4 of them blown up, is that all of these calcium signals are centered on these white, immobile IP3 receptors. You can see that more clearly, perhaps, if I just take a cross-section. So here we're seeing the IP3 receptor shown at the 0 time point in green and at 30 seconds in blue, and you can see they perfectly superimpose. That tells us it didn't move in the 30 seconds. It's an immobile cluster of IP3 receptors. And then when we look at the calcium signals, you can see they perfectly superimpose onto those immobile IP3 receptors. And that's just summarized here. It doesn't matter how we deliver the IP3 to the cell. We can deliver it uniformly throughout the cell by photolysing inactive analog of IP3, the caged IP3, or we can deliver it the way the cell would normally get it through a native signaling pathway by activating the receptor with histamine. The answer is the same. All of the calcium puffs co-localize with immobile IP3 receptors. So that tells us then that actually very, very few of all of the IP3 receptors in the cell are actually licensed to respond. It's a very privileged small subset that are able to respond. Remember, I've shown you that you have to be near the plasma membrane to respond, so that rules out most of the IP3 receptors sitting in the depths of the cell. And now I'm saying that even amongst those subset of IP3 receptors that are near the plasma membrane, it's only the third or so that are immobile that are competent to respond. So it seems then that there is some sort of licensing going on that allows these IP3 receptors to become competent to respond to the IP3 and calcium that will gate them. And we don't know yet what that licensing machinery is. We're actively exploring it. Again, I'm afraid there is stuff missing here. So I'm not on the talk I wanted to be. Let me just, I'm going to have to, there's really a lot of stuff not coming up on this computer, I'm afraid. Let me just summarize the final story for you without showing you the data, because it's not going to come up on this computer. And that is, I mentioned to you earlier, there is a channel in the plasma membrane that will also allow calcium to flow across the plasma membrane into the cell. And that channel is activated by another protein in the ER. It's a protein called STIM. And when the IP3 receptor causes the ER to lose its calcium, not only do we get an increase in calcium in the cytosol, but of course we get a drop in the calcium concentration within the ER. And that drop within the ER is what regulates STIM. It causes STIM to cluster, move to junctions at the plasma membrane, where it is captured by that ORI channel, and the ORI channel opens to allow calcium to flow into the cytosol. And the punchline that I'm not going to have the opportunity to show you is that STIM moves to become adjacent to where those immobile IP3 receptors are at the plasma membrane. So the argument would be, and I think I'm missing even the conclusion slide in this, yeah. So the argument would be, to summarize that then, is that amongst all the IP3 receptors that we have in the cell, only a very small cohort, the immobile IP3 receptors that sit adjacent to the plasma membrane, are somehow licensed to respond to IP3. And those IP3 receptors sit adjacent to the place where STIM will move when we are going to activate that second phase of the calcium signal, the calcium entry across the plasma membrane. So it's an organization that makes sense, but because it puts the calcium release channels adjacent to the place where we are going to have to deplete the ER to give rise to that so-called store-operated calcium entry and activation of that calcium entry pathway. So I'm sorry I can't show you that on a summary slide, since, as I said, it's not showing up on this computer, I'm afraid. What I will do, though, is just then finish by acknowledging the people who have done the work. And again, this is not quite the people that I would acknowledge if I had the right slides. Babu has done all the work on gene editing with a lot of help from Dave and Steve. And not shown on this slide is another very talented graduate student, Pisa Takpa, who did the work on the lysosomes that I described in the first part of my talk. So I'm sorry not for to not be able to show you the movies, which are very pretty, but maybe we can look at those on another occasion. Thank you.
Okay. Thank you, Professor Taylor. That was very interesting talk. I reviewed all my physiology that I took like years ago, and I didn't use them since then. So.